this is an example of a question that is about a sex-linked genetic cross. It says, in humans, a certain rare sex-linked recessive allele, so we're told it's sex-linked, we're told that it's recessive, results in a change of the shape of the iris. This condition is known as cleft iris. Using the symbols capital B and little b, show all the possible genotypes and phenotypes of the children of a man with cleft iris and a woman who was only a normal allele. So the man, that's important because it's sex-linked, is cleft iris, and the female is normal. So we know that the man is XY, female chromosomes are XX, normal allele is B, and we know cleft iris is little b. We've then got the parental genotypes. We've got to write down the gametes. We've got a space here to carry out a genetic cross. And then we've got to state what the offspring genotypes are and the offspring phenotypes. So genotypes is the genes that the parents are carrying. Gametes are the chromosomes and genes that the gametes, the sex cells, are carrying. Offspring genotype is the genes that the offspring are carrying, and offspring phenotype is what is expressed in the offspring. So we have to start off with parental genotype. Right, we know the male is XY, and we know he's got a cleft iris, and we're told that it's sex-linked, which means that you don't find the allele on the Y chromosome. So we're not going to find an allele on the Y chromosome. He's got cleft iris, so it's a little b. So all we do is we write a little b, and I do a dash just to show that there's no gene on the Y chromosome. The female is XX, so she will have 2 copies of the gene, so 2 alleles, and she's got normal alleles. Normal allele is capital B, so she will be big B, little b, big B for genotype. So that's the parental genotypes. Right, the gametes then, we know during meiosis, chromosome numbers halved, so you get 1 copy of each chromosome in the sex cells. So the male is going to carry B, and the other sperm are not going to carry anything, because this part would be of the allele is not on the Y chromosome, and the female, her 2 eggs will either be big B or big B. Right, we then carry out a Punnett square. So we take the chromosomes from the male, so it's big B and dash, chromosomes from the female, or the alleles from the female, so big B, and we draw a Punnett square. And from this Punnett square, we can work out the offspring genotype. So if this sperm and this egg join together, the alleles that will carry is big B, little b. If this sperm and this egg, well, there's no allele on this sperm, so it will just be big B, and I'll put a dash to show there's nothing there. This sperm and egg will be big B, little b, and this sperm and egg would be big B from the female and a dash from the Y chromosome, the male. So our offspring are either big B, little b, or big B dash. So now we've got to say what the offspring genotypes are. So these are the Y chromosomes, X, and this is X, X. So these 2 males, because we know these are males because they've got the chromosome missing, the allele missing off the Y chromosome. So these will be males that are normal. because they're carrying the normal allele. These two alleles on the chromosome, so these must be the female, both the X chromosomes. This is XX, this is XY. So the offspring here, even though these females are carrying the gene for cleft iris, that is not going to show because it's recessive. So these are females. Normal. You could just write all normal because both the males and females are normal. So that's how you carry out a genetic cross that is sex-linked.